In this procedure, the dynamics of gene expression in plant tissues are imaged over time with the use of robotics. First, lima bean seeds are sterilized and placed between moistened paper towels in a tissue culture container for germination. After 4 days, the cotyledons are removed and bombarded with a DNA construct containing a promoter fused to GFP. Cotyledons placed in petri dishes are mounted on a 2-dimensional custom-designed robotics image collection system. The robot is programmed to capture images of each tissue piece every hour for 100 hours. Images are subjected to image analysis to yield quantitative data on the intensity of GFP expression over time.
Hi, I'm John Feiner, the Director of the Plant Transformation Laboratory in the Department of Horticulture and Crop Science at The Ohio State University.
I am Carlos Hernandez-Garcia, PhD student in the Feiner Laboratory at The Ohio State University.
Today, Carlos will show you the protocol that we developed for analysis of gene expression in plant tissues using the green fluorescent protein.
We use this procedure that we have developed in the laboratory for measuring the strength of promoter activity. To do this, promoters are first cloned upstream of a GFP gene, which is our reporter genes. I will show you how we introduce the promoter construct into lima bean cotyledon and send them up on our custom-designed robotic image capture system. I will finish by going over our protocol for image analysis. So, let's go.
To begin this procedure, surface sterilize lima bean seeds using standard procedures. Place the sterilized seeds in magenta GA7 containers between layers of moistened paper towels and incubate them for 4 days at 25 degrees Celsius. Here we use a high copy number expression vector useful for promoter and promoter element